All right. Hi, everybody. I hope you guys are having a good spring break. I went ahead and made this review video. There's still one more class left, but I figured you guys may want to listen to this, for the other half of the break. Hopefully you guys are taking some time to study, but I also hope you guys are taking some time for yourselves. All right, so we are just going to go ahead and jump into it. And we are going to start with enzymes. So this was just so funny. Uh, get it? You know why they call me DJ Enzyme? Because I'm always breaking it down. Hopefully you guys can remember that in the exam. All right, so let's talk about chemical reactions. So what are some ways that we can increase the rate of a chemical reaction? Well, we can do something like increase the temperature, but depending on the organism, this actually may not be an option just because of denaturing that occurs whenever you start to increase temperature. We can also increase the concentrations of reacting substances, but again, this may not be a viable option for the cell just because some of those reacting substances may not be available. It can disrupt homeostasis, and there's also kind of a space limitation. So there may not be enough space in the cell for all of these increasing concentrations. So finally, what we can do is we could add a catalyst. And so a catalyst is a substance that participates in the reaction, and yet it emerges at the end in its original form. And this is probably the most applicable way to increase the rate of a chemical reaction in biochem. And our catalysts are going to be enzymes. And so enzymes versus a non-biological catalyst. So most enzymes are proteins and they're very dynamic. And enzymes are going to contain a specific fraction of the structure where reactions will take place. And this is called the active site. And so what type of amino acids would we expect in the active site? Probably the amino acids that are going to be reactive. Most enzymes also work at mild conditions, so they're going to work at a steady pH, around 7.4, and also a temperature of about 37. The activity of many enzymes are regulated, and so think back to Unit 1 and how we would use enhancers and activators. So we can turn up and down genes being expressed. This is a similar concept to where we can turn up or turn off their activity. So this allows us to have some sort of modulation and also be able to regulate and fine-tune how these enzymes are working. And enzymes can speed up biochemical reactions. So they can speed up reactions from 10 to the 7th to 10 to the 20 times, which is a lot. And chymotrypsin is an example, and it cuts peptide bonds between amino acids. And so the half-life of one of these bonds without an enzyme is 20 years. So 20 years for that bond between amino acids to break. Well, chymotrypsin can increase that to 190 bonds being broken per second. So that just shows you the power of an enzyme. And I also, oh gosh, I hope you guys can't see that. Amanda binds. There we go. All right. So this is actually a protein that I study, and I don't know if you guys actually know this. I think I've said it a couple times, but I'm defending my PhD on the Monday after spring break, so on the 27th, and this little guy is actually what I'm presenting on. So this, we think it's an enzyme, which is really exciting. This unit has been really great for me for my studying. So I study what we think is an enzyme involved in lysine methylation. So we think it's a lysine methyltransferase, and it has this really neat structure that is very similar to this human version of it, this human homolog. And here you can see the substrate bound in the human enzyme, and then you can kind of see where it would bind in our enzyme. So we don't show that it has activity yet, but that's what we're working on. So again, this is just super exciting for me. And super relevant. So you guys can see, scientists even now are working on enzymes, and it's a super cool thing to work on. All right, so kind of like what I just talked about. So active sites, enzymes contain active sites. Probably they're going to be polar amino acids, and they're going to be specific for a substrate, or a reactant that's going to bind. And this is usually going to be excluded from the aqueous environment, so it's kind of going to be hidden. And we need all of the rest of the protein, and that's going to kind of aid as support. So most enzymes are specific for a substrate. And there are specific shapes and amino acids that will bind to our active site. And it's going to have something called molecular complementary. And we're going to have, again, a very specific shape that will recognize these active sites. And there are some enzymes that may do what we call moonlighting. And so these are going to be promiscuous enzymes, and they're going to bind other substrates, not just what we expect. And we're going to talk way more about how these enzymes fit in. And, we'll talk about the lock and key model and all of that in a couple of slides. So we can have 7 major classifications of enzymes. We can have the addition, the removal, the rearrangement. But one exception is going to be a translocase, which we will talk about here in just a second. So the first classification is going to be an oxidoreductase, and these catalyze oxidant reduction reactions, and they add and remove electrons and protons from a substrate. These include dehydrogenases, oxidases, reductases, and peroxidases. Next, we have transferases, which is what we think my protein is. Again, super exciting. So these can transfer a functional group from one molecule to another. So you can have the transfer of an amino, a methyl, again, which is what we think my protein does, or an acyl group. One example of this is actually going to be hexokinase. So if you guys know anything about glycolysis, you'll know about hexokinase. This is also, if you guys know Dr. Jim Morris in the genetics and biochem department, he does a lot of work on hexokinase as a potential drug target for the parasite that we study. The next class is going to be hydrolases. And so these cleave bonds by adding a water molecule. So if you're trying to figure out what's happening, you're going to want to look for water. If you're trying to see a difference, like what kind of reaction is happening, look for water. So again, it's going to cleave bonds by adding a water molecule. These include enzymes like esterases, phosphatases, peptidases, lipases, and glycosidases. Again, look for that water. That's the key for being a hydrolase. Next, you'll have a lyase, and this actually removes a functional group via a non-hydrolytic reaction. And so we often see the result in the formation of a double bond on either side of the equation. So that's what you're going to look for, is that double bond. Isomerases, so these are going to catalyze rearrangements of functional groups within a molecule. You can have a mutase, which is going to transfer a functional group from one position to another. Or you can have an epimerase. Gosh, these words, guys, I'm going to be stumbling this whole time. An epimerase is going to invert functional groups, so flip them. Next, we can have a ligase. And so these use ATP to form bonds between two substrate molecules. So they'll form a carbon-carbon bond, a carbon-sulfur bond, a carbon-oxygen bond. And also, you'll see a phosphate group ester bond. And ATP is used here for energy, and a phosphate group is going to be released. And finally, you can have translocases. And so this is just going to assist in moving molecules across a cell membrane. So a lot of information on this slide. My advice to you for studying these, make some flashcards. Make a quiz and list out kind of the differences that you can see. Hydrolases, you're going to see that water. And then for for transferases, you're going to see, functional groups being added and so on and so forth. So kind of come up with a list of characteristics that are going to be able to distinguish the different classifications. All right, so activation energy. So the activation energy, this is a reaction coordinate diagram, and it's showing reactants to products. And in order to form products, we have to overcome something called delta G double dagger. And so this is the energy of activation. So the top of the peak is going to be the transition state. At the transition state, it's very unstable. So there's no fully formed bonds. The transition state is where the substrate is bound tightly to the enzyme. In my head, I always think about this. You guys know when, think about Alice in Wonderland. When she's falling down the tunnel to Wonderland, things get a little weird in the middle. So she's starting out in London and she's going to Wonderland. That middle kind of tunnel, that's going to be the transition state. So things are just very unstable. It gets a little weird. But then at the beginning and the end, you have two fully formed Alices. All right, so the sign delta G, it just tells us the direction of the reaction, or the spontaneity of a reaction, but not the rate. So delta G is equal to the G of the products, or the free energy of the products, minus the energy of the reactants. And delta G does not consider the activation energy. If delta G is greater than 0, then it's going to be a non-spontaneous reaction. However, if delta G is less than 0, then it's going to be a spontaneous reaction. So delta G does not change with the presence of a catalyst. And I think that this is a typo. So here, so delta G, when it is less than 0, it's going to be spontaneous. Delta G, when it is greater than 0, is going to be non-spontaneous. So this should say non-spontaneous. And again, it doesn't change with the presence of a catalyst. All right, so the next thing we can talk about is going to be the transition state. And so in the transition state, enzymes are going to decrease the energy that is required to get to that transition state. So you can see here, these two different graphs, and that delta G is going to be changed, or I guess just the activation energy is going to change. And so enzymes are going to decrease the energy that's required to get to the transition state, and it's going to decrease activation energy. So we can consider enzymes as matchmakers, which if you guys are in Dr. Anderson's class, that's how she described it. So matchmakers, I always think about Mulan, the whole matchmaker scene. So they're going to bring together two parties to interact. And this is how the activation energy is lowered. So based on the enzyme's ability to bring together the reactants in the proper orientation and proximity, that's what's going to lower the activation energy. So it doesn't lower delta G, just the activation energy. And this is just a really great slide that Dr. Anderson had in one of her PowerPoints, and I stole it. So these are just some really important terms to know. Again, highly recommend making flashcards to be able to identify these. So we can talk about substrate. So a substrate is going to be the substance that an enzyme acts on. So we can think about that as the reactant. We're going to have our products. So these are the products that the substances produced by chemical modification of substrates. So what the substrates become after being modified. Next, we can have the activation site. So this is the region on or within an enzyme where the substrate is going to bind, and that's where the reaction is actually going to occur. So I think that's pretty easy to remember. It's the active site. That's where everything is happening in these reactions. Next, we can have cofactors. And so cofactors are small molecules or metal ions that help and are sometimes required for the catalytic activity, so to make the enzyme function properly. Next, we have the transition state. So this again is that Wonderland tunnel. So it's the unstable or energized intermediate formed in the enzymatic or reaction that has properties of both the substrate and the product. So it's, again, I think the biggest thing here is that it's really unstable, but also it's like the enzyme is bound most tightly at this point. And it's the point of a reaction where the reactants and the products have the highest energy. And next, we have the activation energy. And so this is the energy that's required to produce a chemical reaction. So again, I highly recommend making maybe some flashcards would be great, or I think Quizlet is a thing I've heard of. Again, you guys got to remember, I'm really old. I turned 30 in like a month, so technology is a little hard. All right. So we can have three kinds of chemical catalytic mechanisms that are used by enzymes. So we can have acid-base catalysis. And so this is where a proton is transferred between the enzyme and the substrate. And so as the substrate interacts with the active site, there will be a proton donation. So the enzyme itself is going to donate the proton and allow for a little more stability in that transition state. And a base is going to subtract a proton. In the enzyme itself, and an amino acid within the protein being used is going to be used for proton accepting and stealing. So some amino acids, I think this is kind of the key part here, some amino acids that are going to play a role in acid-base catalysis, that's going to be amino acids like aspartic acid, glutamic acid, histidine, lysine, and tyrosine. And then we can have covalent catalysis. And this is when we have covalent bonds that form between the catalyst and the substrate during the formation of the transition state. And so here, when you have covalent catalysis, you're actually going to have two energies of activation to overcome. And a nucleophile is, and that's going to be in the enzyme active site, is going to form a covalent intermediate with a substrate. And it's going to be looking for an electron pore center. And so what amino acids are going to be involved here? Well, you're going to want to look for amino acids that can give up a proton around, a physiological pH of about 7.4. And then we can have metal ion catalysis. And so metal ion will mediate oxidation reduction reactions or promote the reactivity of other groups in the enzyme's active site through electrostatic effect. All right, so here is where I was talking about kind of how enzymes fit together that we would talk about later. So one model that was proposed is going to be the lock and key model. So we know now, science is all about learning, and you can produce a hypothesis or a model, but that's always going to change. That's the beauty of science. And so we know now that this model is not true, but when it was proposed, it was one of the best models we had. And so what we know now is that the lock and key model actually describes the transition state. And so this model was proposed to suggest that the substrate is precisely aligned in the active site, and it fits the enzyme like a lock and a key, or a key in a lock. Enzymes, like other proteins, are not rigid molecules, but instead can, and they also must flex while binding their substrates. So, again, here you can see it fits perfectly. We know that's not the case anymore, and things can actually shift around and form around it. So that leads us to the induced fit model. And so this is, you can see here, I think this is a great picture showing, you have a general form, but then it actually molds to fit the enzyme or the substrate shapes. And so in the induced fit model, when a substrate binds, the active site is actually going to form around it. And again, remember when we're talking about proteins, proteins are so dynamic. And so what reactions are being interrupted when a substrate binds in this model? So we're going to be interrupting certain bonds like hydrogen bonds, hydrophobic effects, and van der Waals forces. But again, you can see in the induced fit model, your active site is going to form around your substrate. And I thought this was a great little comparison between them. So here you have the lock and key model on the left, and on the right you have your induced fit. So again, in the lock and key model, your active site fits exactly around the substrate. You're not going to have any alteration to the structure of the enzyme, and it's a less accurate model when we're talking about enzymes. But for induced fit, you can have the active site and it's going to actually mold itself around the substrate. And that's only when the substrate is present. The structure is going to be modified a little bit for that enzyme to function, and it is a more accurate model of how enzymes actually bind their substrates. And so enzymes are going to stabilize the transition state. And so in the transition state, we are breaking the old bonds and making new bonds. And here we're increasing the rate of the reaction because it takes less activation energy to get to the transition state. And so the lower the activation energy is in getting to the transition state, then we're able to increase the rate of the reaction. And I do want to point out, I have this in all caps in my notes. The transition state is where it binds most tightly. So remember that. Enzymes are also going to rely on proximity and orientation effects. And so if we come back here and look at the active site, so just keep this in mind, the way the active site accommodates the substrate is going to be specific, and it has to be in the correct orientation to have a reaction occur. And so one enzyme that we can think about is going to be chymotrypsin. And so chymotrypsin is a serine protease. And so serine proteases have a triad of amino acids in their active site. And we do probably, hint, hint, wink, wink, need to know the amino acids in the triad. So they are important in the reaction. And there is a serine in the active site and a protease because it cuts peptides specifically around the aromatic structures. It's going to hydrolyze peptide bonds adjacent to the aromatic amino acids, and it cuts peptide bonds linking those aromatic rings. So it's going to help break down proteins in the digestive tract. So chymotrypsin, it's going to help break those proteins down so that we can absorb their nutrients. And now we're going to get to the catalytic triad of chymotrypsin. And so these serine proteases, they use covalent catalysis and acid-base catalysis. And so in that triad, there's a serine, and that's going to play a role in breaking those covalent bonds. There's also a histidine, and the histidine is going to play a role in the acid-base catalysis, and it's going to donate or accept protons. And then you're going to have an aspartic acid that is more of a support amino acid that's going to hold it in place. So in many organisms, the activity of proteases is limited by the action of protease inhibitors. And by synthesizing the proteases as inactive precursors, we can kind of get around this. And so we can use something called zymogens. And so zymogens are going to be later activated by proteolysis. And so again, we don't want these enzymes working before they get to the spot where they need to be to function. And so we can have them created as zymogens. And so for chymotrypsin, these zymogens are going to be secreted into the small intestine. And then when it gets into the small intestine, trypsin is going to be activated first, and then it will activate chymotrypsinogen through cutting out small areas in order to activate it. And so here, the big thing to know is that trypsin is the key. So it's going to have proteins that are going to protect it while it's traveling throughout the intestinal system, and it's going to protect it from being activated early. So what happens if trypsinogen is activated early? Well, it's going to unlock everyone early, and it's going to lead to things that you can think of. Again, if you're going into the medical field, it's going to lead to things like pancreatitis, and that's where proteins are being broken down too early. And so that was a lot of information. So we're going to take a little mental break. Here are, you guys know I can't show you a PowerPoint without showing you pictures of my dogs. And so these were just two cute pictures I took this weekend of Honey and Barley. Barley looks like he's always anxious, so and Honey likes to sit there and model. All right, so now let's get into enzyme kinetics. And so kind of a chemical kinetic overview. So whenever we're thinking about kinetics, we can think about thermodynamics and think about delta G. And so this can predict whether a reaction is going to be spontaneous, but again, it's not going to predict the rate. So the rate or velocity of a reaction is the change of a concentration of a reactant or a product per unit of time. So here we need to know an amount, and we also need to know the time. So reaction velocity can be thought of as concentration versus time. And over time, we can have a negative view where the substrate is decreasing and a product is increasing over time. So a positive would be a product increasing over time. A negative is where a substrate is decreasing. And so we have a formula, and it's V equals K A. And so this is going to be where A is going to be the concentration of either the substrate or of the product. And so how does this rate relate to the substrate or the enzyme that I have? Well, we can think about the progress of an enzyme catalyzed reaction. And so whenever you have less enzyme, so here we can think about it. So whenever you have less enzyme, your reaction is going to be slower. But whenever you have more enzyme, you're going to have a faster reaction. So the more enzyme you add is going to increase the rate. It's always going to increase the rate. And you can think about it, again, Dr. Anderson explained this really well in her class, but you're going to have more machinery to make the product that is needed. So if you think about it as I'm blanking, as like a work line, you're going to have more of those machines. Like think about it as building a car. If you have more machines to build cars, then that's going to obviously make the process go a lot faster. So if I add more substrate, but not more enzyme, then the rate is not going to increase, right? You can have so many parts, but if you don't have the machines to actually put them together, then you're not going to increase the rate. So that's what we call saturating the system. So enzyme kinetics is a branch of chemical kinetics. It can give us information about reaction rates and is the quantitative study of enzyme catalysis or enzyme kinetics. Kinetics can also measure enzyme affinity for substrates and inhibitors. And we can answer questions like, what is the affinity for a substrate? What's the affinity for an inhibitor? And we can talk about things in terms of order. So order is useful in describing reactions, and it is determined experimentally. So we can look at first order, which I have here. And so the first order is unimolecular. So the rate is only dependent on one molecule and how much of that one molecule is around. Second order is going to be bimolecular, and so it's dependent on two molecules interacting. And then zero order is when the rate is not affected by adding more substrate. So this at zero order is what we call the system being saturated. And so here we have velocity versus substrate plots. So the enzyme concentration is going to be held steady. The y-axis is going to be the velocity, and the x is going to be the amount of substrate. And again, the enzyme concentration is the same in this plot. So you're going to have saturation. Again, we have too many parts at this point and not enough machines, and so you're going to saturate the system. So we can actually describe these processes by mathematically, and we can use the Michaelis-Menten kinetics for this. But I do want to point out that not every enzyme follows Michaelis-Menten kinetics, but most will. So enzyme and the substrates are going to be interacting in forms K1, so you can see that here. And this is going to be a first order reaction, and it can also be reversed here. The enzyme and substrate complex is then going to form and product at K2, first order, and it's going to be normally able to be reversed or not normally able to be reversed at this point. So K2 here is going to be the rate limiting step. So the velocity is the change of product forming over time, and that is equal to the rate of the enzyme substrate complex. As the substrate increases, the kinetic behavior can change from first order to zero order in kinetics. Again, remember, we're saturating that system. And this equation will be provided on the exam. And so all of this is related to rates of the substrate concentration and then of product formation. So V0 over here, so this is going to be the initial velocity. So the velocity at the start of the reaction. And it's the rate of the linear portion, so of this first order reaction. The Vmax is going to be the maximal velocity, so you can see that right here. So this is how quickly a product can form. And then you're going to have something called Km, and Km is going to be the Michaelis-Menten constant. And so Km is substrate concentration at half maximal velocity. So at half Vmax, that is where you're going to have the substrate concentration, and that's going to be our Km. So a small Km means a high affinity of the enzyme for its substrate, and I might have said that wrong. A low Km or a small Km means a high affinity of the enzyme for its substrate, but a high Km means low affinity. So just kind of keep that in your brain as it's opposites. So a small Km, you're going to have high affinity. A high Km, you're going to have low affinity. And this tells us how much it likes a substrate. So how much the enzyme actually likes its substrate, or it's, again, that biochem term is going to be affinity. So Vmax, this is how much product I can make per unit of time. So Vmax is the max velocity of a reaction, and the max velocity of a reaction that it can attain, and it is not a constant. So why is it not a constant? Well, it depends on how much enzyme there is, or enzyme concentration. We can also talk about kcat. So kcat is going to be a measure of catalytic activity. And so this is the Vmax divided by the total enzyme concentration. So this kcat tells us how efficient it is for one molecule of enzyme. And so values can range from less than 1 per second to many millions per second. So whenever we're thinking about kcat, a high kcat is going to be more efficient. We can also look at kcat over Km. And so this is what's going to indicate catalytic efficiency. It is an estimate of how perfect the enzyme is, and it measures how well the enzyme performs. So a large number here is going to be more efficient. And so there's a limit for the kcat and Km, and that's called the diffusion limit, and it's about 1 × 10⁸. And this is the rate at which the enzyme and the substrate will diffuse together. All right, so above that, so above about 10 to the 8th, it's not like perfect. So again, for this, for kcat over km, remember that it's an estimate of how perfect the enzyme is, and it indicates catalytic efficiency, and a larger number is more efficient. All right, so we can then move to Lineweaver-Burke, and so this is going to linearize the Michaelis-Menten kinetics data. And so if we look here at how we get this equation, you take the reciprocal of both sides of the Michaelis-Menten equation, and you end up with what looks like the equation of a line. So we can think of y equals mx plus b. And the km over Vmax, so this is going to be our slope. Our 1 over Vmax is going to be our y-intercept. And the Lineweaver-Burke here in the graphical form, our x-intercept is going to be -1 over the km. And so again, not every enzyme is going to fit Michaelis-Menten kinetics. An example of this would be for 2 substrates. So Michaelis-Menten is only going to be for 1 substrate, but some enzymes have multiple. And actually more than half of the known enzymes, they actually have more than 1 substrate. And so this is done through like cooperative behavior. So we can think about hemoglobin. You can have an active site and also an allosteric site. And so the allosteric site is a site other than the active site. And it's going to have a place for more than 1 type of small molecule to bind in order to get activity. And so these are cooperative behavior enzymes are going to give us something that looks like a sigmoidal curve. So again, that's where you need 2 or more different molecules to bind in order to get enzyme activity. And so we can also think about how do we inhibit enzymes? So what does it mean to inhibit? So whenever you're inhibiting, you're going to turn off activity. And the best way to think about this is how drug therapeutics work. So some enzymes work in an irreversible manner, and these inhibitors bind covalently to the active site. We can also call these suicide inhibitors. And so one example of this, you can have deoxyribose 5-phosphate or DUMP, and have it this thymidylate synthase going to deoxyribose 5-phosphate or DTMP. And so here this fluorouracil is going, this is the inhibitor, and so it's going to bind to the dump, and then we'll stop the process of cell division and replication. And this can be used for chemotherapy as a way to stop cells from rapidly dividing. Another example of this can be penicillin. And so this one is an irreversible inhibitor of the enzyme glycoprotein that catalyzes bacterial cell wall synthesis. And so whenever you have an irreversible compound, you don't have to consider the kinetics because there's nothing to measure. You just turn off enzyme activity. However, we can have competitive inhibitors. And so these are going to be inhibitors that bind the same site as the substrate. And so here you can see our substrate looks like a pizza slice and it's going to bind in there, but our inhibitor, it also looks very similar to a pizza slice. And so it can compete with the substrate to bind in that active site. And so competitive inhibitors, they compete with the substrate to bind to the active site. They're going to increase the Km, but not affect Vmax. And so that is really important whenever you're trying to identify between the different kinds of inhibitors. Competitive inhibitors increase the Km, but do not affect Vmax. And so they're going to decrease the affinity for the substrate. So you can overcome competitive inhibition, and you can do this by increasing the substrate concentration. Ibuprofen is an example of this. It's going to be a competitive inhibitor. And here, whenever you're looking at the Lineweaver-Burke, if the y-intercept is changed, if the y-intercept is unchanged, but the x is changed, so if the x moves more towards the origin, then this is going to be a competitive inhibitor. And the transition state analogs here, whenever you're thinking about inhibitor design, if you can make an analog that resembles that transition state, then that's going to be a better inhibitor. And remember that the transition state binds so tightly, and so inhibitors that resemble that are going to be preferentially bound. So again, the biggest things off of this slide are going to be competitive inhibitors. They're going to increase the Km, but not affect the Vmax. If you're looking at a Lineweaver-Burke plot, the y-intercept is unchanged, but your x is going to move towards that origin. So we can have something called pure non-competitive inhibitors, and these are going to decrease Vmax. And so here the inhibitor binds to a site that's other than the active site, and it can bind either just the enzyme or the enzyme and substrate complex. And it will inactivate any enzyme that binds to the inhibitor, making it less effective. So here, the things that you want to look for is that the Km is unchanged, so the affinity has not changed, but the x, whenever we're looking at Lineweaver-Burke, the x-intercept is going to remain the same. You can see that down here. Again, that Km, but the y-intercept is what's changing, so your Vmax is going to decrease. Next, we can have mixed inhibitors. And mixed inhibitors are actually going to affect both Vmax and Km. And so it's rare for Km to not change. So pure non-competitive will be rare. You're probably either going to see an increase or a decrease. But here for mixed inhibitors, you're going to see that Vmax will decrease, and also you're going to have a change in that Km. And so the big things, if you're looking at Lineweaver-Burke, what you're going to want to look for are that neither x nor y intercepts are the same. And then you can have something called an uncompetitive. These names are horrible. So an uncompetitive inhibitor, and this is going to reduce the Km and the Vmax by roughly the same amount. And so it's only going to bind the enzyme substrate complex. And so here, what you're going to want to look for on the Lineweaver-Burke plots are going to be parallel lines. These are extremely rare to find, but again, you're going to have Vmax and Km reduced by about the same amount, so you're going to end up with parallel lines. This is a really horrible table, so I'm sorry for that, but hopefully you can see it. I thought this was a great thing to add in and a great study tool, but I think what you can do, I think I just Googled it and searched for like kinetic characteristics of reversible inhibition, and I found this table. And so it's showing you that competitive, the type of inhibition, the Km is going to be higher, your Vmax is going to be the same, and your slope is going to increase. For uncompetitive, you're going to have a lower Km, a lower Vmax, and your slope's going to stay the same. And then you can have the non-competitive. Pure is going to be Km stay the same, Vmax be lower, your slope is going to increase. In mixed, you're going to have a higher Km, a lower Vmax, and an increase in slope. So here you can really see the differences and kind of what to identify on an exam. All right, so these are just some mechanisms for regulating enzyme activity. So you can regulate the rate that the enzyme synthesis and degradation is occurring the enzyme's location can aid and regulate the activity. So remember, we talked about chymotrypsin and kind of how it moves through the system and is then activated. Chemical signal that can help and regulate enzyme activity. And you can also have these modifications to enzymes. So you can have, here, for example, the addition of a phosphate or the deletion of a phosphate group to activate or inactivate your enzymes. All right, so that was a lot of work. And I do want to point out there are some math problems that go with that, but again, I know I ranted about it last time I made a video, but I just don't have the capabilities to kind of draw that out. But I've talked with a few students, and one of the best things I came up with was whenever you're going through your math problems, write out the steps. You know, uh, if you're trying to find KI, for example, I think that was a homework question. Write out the steps for how you did that. So, the first step, you would want to use like the no inhibitor line to solve for Km. You then use the positive inhibitor line to solve for Km apparent. You then solve for alpha, and then you then solve for Ki. So breaking down these mathematical problems into steps like that, I think makes it a little more, it's a little easier to remember how to do that on an exam. But my biggest advice or suggestion is to go back through and practice all of those problems. How do you find Km or Ki? And then there are a lot of resources online as well. I did go through and I looked through YouTube just to see like how many videos are out there. There's a ton of videos out there for enzyme kinetics. So again, I highly recommend doing that. Practice, practice, practice. That's going to be the best way. Practice your problems in your book, practice your homework problems. I know that in class activity, that's also going to help a lot as well. So for our mental break, I have baby puppies. So this was Honey the day we got her from the Humane Society. She was a little potato. She was so cute. We think she was actually like 4 or 5 weeks old. She was really little. Barley, we got him when he was a little older. So this was actually the day we got him as well. So he was, I think, like 5 or 6 months old at this point. Um, and then just because I realized I'm being pretty heavy on the dogs, this is my parents' cat. We weren't really cat people, but I know there are some people who are. So her name is Minerva and Kitty Kitty. So that is for the cat people. All right. So now we're going into our final section of carbohydrates. And so we can do a carbohydrate overview. So what are carbs? Carbs are the most abundant biomolecule in nature. They have a wide variety of cellular functions. They can be used for energy, structure, communication within the cell, or also as precursors for other biomolecules. They're a direct link between solar energy and chemical bond energy. And so we can break them down into monosaccharides. So monosaccharides are going to be the simplest biological molecule. And these are basically going to be carbons that are hydrated. So you can see that here. And so they're going to be typically constructed of carbon, hydrogen, and oxygen atoms. You can have carb derivatives that have other groups on them, like nitrogen. And monosaccharides can form what we call an aldehyde, or they can be made from aldehydes, and they're going to form aldoses. Or you can also have a ketose. And so an aldose is going to be the carbonyl carbon, so the carbon bond with a double bond here on the oxygen. So that's when it's going to be on the first carbon. So an aldose is going to have that carbonyl carbon on the first carbon. A ketose is going to have that carbonyl carbon on the second carbon. And so in nature, we don't really see it on the third or even fourth carbon. So what you're going to want to look for is that second carbon for identifying a ketose. And so one kind of way to think about it is an ose can indicate a sugar or a carb. And so these can be classified by the number of carbon atoms that they contain. So for example, we can think of pentose and hexose. A ribose is going to be an aldose with 5 carbons. And then a glucose is going to be an aldose with 6 carbons. And another name for this could be an aldohexose. Another name for this would be a pentose or an aldopentose. Again, we can look at where the double bonded oxygen is. And here, this is fructose, so this is going to be a ketose, and we can, again, look at where that double bonded oxygen is. And another name for this is going to be a ketohexose because you have 6 carbons and it's a ketose. And so we can also identify chiral carbons. And so a chiral carbon is going to be the carbon atom that has 4 different groups around it. So nearly all monosaccharides have a number of stereoisomers, and that's easy to see whenever they're in these Fisher projections. And so here we can identify the chiral carbon and move from there. So stereoisomers, they're going to be molecules that differ from each other in only their configuration. So whenever we're talking about configuration, we're talking about the 3D arrangement of groups around that chiral carbon atom. So for example, glucose has 4 chiral carbons. And so we can come up with this number by having 2n. And so 2n, n is going to be the number of chiral carbons. So this means that for glucose, you have 4 chiral carbons. So we'll put a 4 here. And so that means glucose is going to have 16 stereoisomers. The simplest ketose does not have any stereoisomers. And from here, you can further classify it, which we'll get into. So enantiomers, they're going to be a non-superimposable mirror image. So glyceraldehyde is going to be the reference here. And you can see down here. And so if the hydroxyl group is to the left, then it's going to be an L enantiomer. And then if the hydroxyl group is to the right, then it's going to be a D. So in nature, most of the sugars are going to be of the D enantiomer. But if we have more than one hydroxyl, then you're going to number your carbons starting with the highest. And so here you can see the numbering. And these are also examples of diastereomers. So these are not going to be mirror images. So mirror images, not mirror images. You can also have a cyclical structure of sugars. And so sugars with 4 or more carbons are actually going to exist primarily in a cyclic form. You can have 5-membered rings, and these are going to be called furans or furanoses, and a 6-membered ring, which is going to be a pyran or a pyranose. For example, the cyclical form of fructose is going to be fructofuranose, while glucose is a pyranose, and it's going to be glucopyranose. And so hexoses and pentoses can actually form planar structures when the sugar ring it kind of puckers. So each carbon atom can then retain its tetrahedral binding geometry. So if we think back to organic chemistry, we can think about those chair conformations. So you kind of have to think about these in a spatial way. So in order to have those binding patterns and the geometry of that, it kind of has to bend or pucker. And so the substituents of each carbon, so whatever's bound to each carbon, can either point above the ring, which is going to be called axial, or it can point out of the ring, which is going to be equatorial. And so the bulky hydroxyl groups in the equatorial position, that's what's going to lead to stabilization. Glucose can adopt a chair conformation, And that's when all of its bulky ring substituents, so these groups, these red groups, those are going to be the bulky ones. They're going to be occupying equatorial position, and that's going to minimize the repulsions between neighboring groups. So it's going to kind of help keep these away from each other. In other hexoses, some of these groups may occupy the more crowded and be less stable axial positions. So the greater stability of glucose may be one of the reasons that it is abundant among monosaccharides. So again, think about the position of these different groups is going to lead to stabilization. Glucose is really stable because it has all of these different groups into the equatorial positions. And so, unlike enantiomers and epimers, which are interchangeable, anomers in an aqueous solution can freely convert between the alpha and the beta groups. And so if the anomeric carbon is modified, then the sugar is going to be locked. So you can see that here, or not here, but it's going to be locked and it's going to become a non-reducing sugar. And so, phosphorylated monosaccharides, you can have phosphate groups added. So sugars can have phosphates, and that's going to aid in increasing energy. And so they're named by where the phosphate is added. So here we can see glyceraldehyde 3-phosphate, so it's on the third carbon. Here we see fructose 6-phosphate, which is going to have a phosphate group added to that 6th carbon. You can also have nucleotides or nucleic acids added to monosaccharides. And so here we have a ribose, which again is an aldose with 5 carbons. And you can have that carbon added with a base. And so these are now going to be considered non-reducing sugars as well. And so you can also have polysaccharides. And so polysaccharides are a carbohydrate chains that are called glycans. And so each monosaccharide has several free hydroxyl groups that can participate in a condensation reaction. And this is going to permit different bonding arrangements that allows for what we can call branching. And so this is going to involve the anomeric carbon for the formation of glycosidic bonds. And it's going to be limited, but in theory, all hydroxyl groups could be reactive. And so here we can think about lactose. So some of you guys may be lactose intolerant, but lactose is a milk sugar, and it's the disaccharide that's found in milk. So one molecule of galactose will link to one molecule of glucose in a beta 1,4 linkage. So you can see that here. It's common to have a deficiency in this enzyme that breaks down lactose, which that enzyme is called lactase. And lactose is also considered a reducing sugar. Sucrose. So this is going to be a common table sugar. It's where one molecule of glucose is linked to one molecule of fructose, and it's linked by an alpha beta 1,2 glycosidic bond. And this is going to occur between both anomeric carbons. And so sucrose is a non-reducing sugar. So if kind of the key for if it's in this format to look for a non-reducing sugar, if you see 2 Greek letters, then it's going to be a non-reducing sugar. All right, so starch and glycogen. Starch and glycogen are polymers of glucose only. And only glucose. So starch is what plants use for storage. So you can have amylose or you can have amylopectin. So amylose is a linear molecule shown here that connects the glucose through an alpha 1,4 linkage. And amylopectin is going to be a branched molecule, and it is going to have linear linkages, so an alpha 1,4 linkage again, similar to amylose, but you're also going to have these branches that are an alpha 1,6 linkage. And these branches typically form about every 18 to 24 glucose molecules. You can also have glycogen, which is going to be branched a linear alpha 1,4, and it also branches at an alpha 1,6. And these are more highly branched than amylopectins. So another one that we can talk about is cellulose. So cellulose is composed of glucose, and it's going to be linked by beta 1,4 linkages. And so, whereas starch molecules form compact granules inside of the cell, cellulose is actually going to form and extend fibers that aid in rigidity and also strength to plant cell walls. And so, cellulose is a linear structure, it's not branched, and individual cellulose polymers can form bundles, and those bundles are going to have hydrogen bonding within and also between the adjacent strains. So that's how plants get those the cell wall and strength to that cell wall. So chitin is going to be the exoskeleton of insects and crustaceans, and the cell walls of many fungi. They can contain a polymer called chitin, and chitin is going to have beta 1,4 linked residues of a glucose derivative in acetylglucosamine. And so this is a glucosamine with an acetyl group that is linked to its amino group. And you can see that here. So these are chitin, and then this is cellulose. And that's how you tell the difference is they're linked very similarly, but chitin again is going to be a glucose derivative with that acetyl group linked to its amino group. And here, this is just a table I found that you guys can feel free to pause and look at, explaining the differences in chitin versus cellulose. So then we can talk about glycoproteins, and these are going to be in-linked proteins. Most of the proteins are secreted from eukaryotic cells or remain on their surface as glycoproteins. So glycoproteins are in-linked via asparagine or O-linked via serine and threonine. And the amino acid sequence or local structure of the protein, as well as the processing enzymes present on the cell wall, are going to determine which sugars are added and deleted from glycoproteins. And so it's important to know that under the adding of the sugars, or what we call glycosylation, occurs during translation, or if you guys remember, protein synthesis. And so glycoproteins are really important for a few different reasons. So one of the biggest ones is that they provide identification for the cell. You can think of glycoproteins as kind of a barcode. And so one of the best known and also clinically important carbohydrate classification schemes using this model is going to be the ABO blood group system. And so that involves oligosaccharides attaching to sphingolipids and proteins on red blood cells and other cells, and that's how we get different blood types. You can also have proteoglycans, and so proteoglycans are glycoproteins in which the protein chain serves mainly as an attachment site for some enormous linear O-linked polysaccharides that are called glycosaminoglycans. And these can be membrane, transmembrane proteins, or they can also be lipid-linked. The function of these proteoglycans is going to be structural. So it's going to be involved in connective tissues, and it's going to attract various amounts of water based on mechanical pressure. So they have a sponge-like action of the glycosaminoglycans in the space of the joints, and that's going to be really important for providing shock absorption. And so proteoglycans can also be known as mucins, and these are going to help in forming the protective mucus lining in the respiratory, gastrointestinal, and reproductive tracts. And so next, this is is where the classes may differ. And again, I forgot to put it at the beginning, but please remember that my reviews are not all including. There's a lot of material to go over for these exams. So I do the best I can, but please remember these reviews are not all inclusive and all including, but hopefully they give you a little bit to go off of. And so there's still one more class after this review comes out. So I'm not sure how far each section is going to make it into metabolism. So I'm just going to give a basic overview. I don't have many slides on metabolism, just because I'm not sure exactly where each section is going to stop with that material. So remember, whatever's covered in your class is free game for the exam, but it may not be covered in this review. So just an overview of metabolism. You can have a catabolic, which is going to be a breakdown of molecules. And you can think about, again, I keep going back to Dr. Anderson, but cats tear things apart. That's one thing she said in class. Yeah, I know my parents' cat, she likes to shred the couch. So catabolism is going to break down molecules, cats tear things apart. You see a release of energy. And it's going to break down into building blocks and then build something else. And so then you can have anabolic reactions, and this is where you're going to be synthesizing molecules, and this is going to use free energy. And so digestion, we can think about these processes in terms of digestion. So we take polymers that are broken down into monomers. And so polymers to think about in digestion are going to be proteins. So the monomers for proteins are going to be amino acids, and those are going to be peptide bonds that are broken. And the enzyme that's going to break those down are going to be proteases. Lipids, so they don't form polymers, but we can think about like triacylglycerols. And we will call the monomers fatty acids and glycerols, and those are going to be linked by ester bonds that are then broken by lipases. And then you can also have polysaccharides, for example, amylose or amylopectin, and these monomers are going to be linked by glycosidic bonds, and they're going to be broken down by amylase, which is going to help start that process. Starchy foods are going to be hydrolyzed by amylases. So amylase is a salivary enzyme, and it's going to break down starch, and that's going to consist of linear polymers, of glucose residues, and then they can also be branched as well. Monosaccharides can enter the cell via active transport, and then and that's done through what we call the glucose transport system. All right, and so for transport, just to go back to fatty acids, those can often be linked to cholesterol and packed into what we call lipoproteins. And inside the cell, the triacylglycerol digestion products can reform triacylglycerol. And here, remember that the lipids are hydrophobic, so they can't travel through the aqueous environment in the bloodstream. And so that's why they can be linked to cholesterol and packed into these lipoproteins in order to be transported to the liver. And so after breaking down into monomers and transporting to the liver, and again, remember the liver is going to be the main organ of metabolism, our body can either choose to use or store this energy. And so the liver is going to help them decide. And so when would we use them? So we would use them when there's not a lot of energy available, and they're going to be used directly for energy when we're low. But if we have excess energy, then it can be stored. And so this is where we are going to end this review. Please remember that there is one more class to cover material in, and I'm making this video early, so I'm not going to be able to cover that for you guys. But just keep in mind, whatever's covered in class is free game. This is not a comprehensive review. Also, good luck to you guys. I hope you guys are having a good and safe spring break. Take time for yourselves. but also, hopefully you guys are studying a little bit. And I also want to point out that I am not having office hours on Monday, the 27th. That is when I will be defending my PhD. So, I will not be having office hours that day, but I will be having them on Wednesday, the 29th. So feel free to email me. I probably will not be responsive Sunday or Monday, but again, I will get to your email as soon as I can. And I will see you guys next week for the exam.